Hi, Michelle Glass here. Welcome back to our second episode of Chapter 3, Lecture 3 series. And our topic for this video is the cell membrane. We spent our last video going through the molecular structure of the cell membrane, and we also described its overall job as regulating the entry and exit of materials in or out of the cell itself. Because remember, our cell membrane is that outermost border of the cell. There are three different mechanisms to describe how material will move across the membrane. So we're going to be talking about the process of diffusion. We're going to talk about a special type of diffusion called osmosis. That will have its own video presentation. And then we will also talk about vesicular transport as our third mechanism of how materials can move across the membrane. So let us begin with diffusion. Now there are some basic descriptions for diffusion that we need to know, and then there are some special types of diffusion that we need to discuss. As we talk about diffusion, we are talking about the movement, so here's our definition, the movement of solute from a high concentration to a low concentration. So diffusion, again, is the movement of solute from a high concentration to a low concentration. Now, we've defined the term solute already. I like to talk about it as the stuff that is being dissolved. So in the case of the cell, solutes would be ions. They would be nutrients like carbohydrates, lipids, proteins. They can be gases, oxygen, carbon dioxide gas. When we talk about movement from a high concentration to a low concentration, we can refer to this as our concentration gradient. And I like to abbreviate that in this way, as you can see up here, where my brackets are representing concentration. So this is kind of my shorthand for saying from high solute concentration to a low solute concentration. And then again, sometimes I'll just refer to this as the concentration gradient. What I like to imagine is that this is my hill. So my high concentration gradients where I have the most of my solute, and then my low concentration is where I have my least amount. When we are talking about diffusion again, we're going to be referring to both solute and solvent. So we do need to make sure we're understanding these terms. In most cases, our solvent is referring to our water molecules. Remember, water is our universal solvent. The definition of the term solvent is the dissolver. And because we're talking about the chemistry of a cell, this is chemistry that's either in the ICF or the ECF, both of which are aqueous or watery-based solutions. And so in those cases, the solvent is water molecules. Solute, remember I said, is this stuff that's dissolved. So if we're using an example of like a lemonade mix, you add in that powder, you pour in your water, and when that lemonade is stirred and dissolved, then you have what is referred to as your solution. So when we're talking about diffusion, we're talking about the movement of the stuff or the solvent, the stuff or the solute in our solutions, okay? And so how are we getting materials to dissolve in that solution? And that is our discussion of diffusion. So remember again, materials are moving with their concentration gradient from a high concentration to a low concentration. So let's look at a more specific example. Okay, so here I'm showing my concentration gradient as a hill, and I like to use my example of, riding your bike down the hill, because just like it takes no energy for you to ride your bike down the hill, it takes no energy at all for your solute to move from a high concentration to a low concentration. Just like gravity will pull you down the hill and it's an automatic process, the only way to stop yourself is to apply energy in the form of brakes or construct a physical barrier to keep yourself from rolling down. Your solute will automatically, without the use of energy, move from a high concentration to a low concentration. And the only way to stop it is, again, to apply energy or construct a physical barrier, which is really the role of our cell membrane. And this, just like your bicycle, is a passive process, meaning no energy is required. Of course, in this case of the cell, energy is called, our energy molecule is ATP. So when we're talking about simple diffusion, when we're going with our concentration gradient, this is a passive process, no energy is required. So this is what we describe as simple diffusion. So when you're pouring in your lemonade mix in with your water, and you see that the lemonade mix or the solute is going to dissolve in with that water automatically, this is diffusion. You are anxious to drink your lemonade, so you stir it up, which is going to increase that rate of diffusion, but you wouldn't have to stir that in order to get that mixture to occur. Another type of diffusion is described as channel-mediated. Now, we talked about channels when we talked about our cell membrane anatomy. So remember, we saw proteins embedded in the cell membrane forming these specialized doorways. Now, in the case of your channels, there are two different types. There's what's described as a leak channel. Leak channels, by definition, are always open. So that means that material is always able to move in or out of the cell using that doorway. Then we have what are referred to as gated channels, and these can either be active or inactive. The drawing that I have for you right here, nothing can squeeze through, so I have a drawing here of an inactive protein channel. A lot of times I like to refer to the active as activating or opening the channel, and then inactivating or closing the channel, just because we have this idea of a gate, and we know that gates can open and close, and so I think that that is a good sort of way to discuss those. So you might hear me use those terms in the future. Active and inactive is a really more correct way to describe what's happening there. Now, this is still a passive process because we still see that our polar molecules and our ions are still moving from their high concentration to a low concentration. So again, because we're going down the hill, then this is a passive process. No ATP is required. So again, you're going to see your channel-mediated diffusion for those molecules that are polar, or those ions that are not able to simply cross through the phospholipid bilayer. The third type of diffusion for us to talk about is what's described as carrier-mediated. Carrier-mediated still involves proteins, but these are a little bit more complicated than a straight-up channel, which can either be open or closed, activated or inactivated. In the case of carrier-mediated, there's actually two types. So let's look first at facilitated diffusion. That term facilitate means to help. So facilitated diffusion is diffusion with help. In this case, our helper proteins are going to be involved in moving really big molecules. So maybe glucose is going to get through via a carrier protein instead of a protein channel. So protein channels, what we saw a minute ago, so let's go back. Protein channels are used for small polar molecules, so like water, and ions primarily. And then our facilitated diffusion involves larger molecules such as carbohydrates. So glucose is going to be an example of something that's going to use a carrier protein. Now in this case, what we see is those polar molecules have to actually bind to a protein. When that molecule is binding, it's going to change the shape. Oops, I did not mean to get that so big. How do I make it smaller? Eek. Okay, oh, right here, I guess. Yep, sorry. And so we're seeing that when the molecule binds to our carrier protein, that's going to cause it to change shape. So it can either open up into the inside of the cell to deliver that molecule, or it can open up to the outside of the cell in order to get that molecule to exit. Here is a nice diagram from your textbook showing the glucose molecule binding in with the carrier protein, and then that's causing that carrier protein to change shape and deliver that glucose into the inside of the cell. Okay, now we said there are two types of carrier-mediated diffusion. We had facilitated diffusion, which used a carrier protein, and now we're going to look next at active transport. It's also going to be using a protein, but also it's active, which means it's going to require some ATP. We have this little note here that both types of carrier-mediated diffusion use integral proteins. The term integral means integrated, so that means it's actually a part of, it's embedded into that cell membrane. So let's look at active transport. The very name active tells us that ATP energy molecule is required. So everything that we've looked at before this is passive. Here is our first type of active diffusion. In the case of active transport, it is a signature that we are going from a low concentration to a high concentration. So because energy is applied, we can actually go uphill. The importance of active transport is that the cell gets to regulate which direction the ions or solute moves instead of just diffusion being involved in that. When you are moving against the concentration gradient, then energy is required. Active transport doesn't always have to go against the concentration gradient. It, by definition, just means that it requires ATP energy, but the only way to go against concentration gradient is to use ATP energy. The most important example for us in this course of active transport is what's described as the sodium-potassium exchange pump. And it's nice that we have this term pump because we know to pump something requires some energy. So we are seeing a protein involved here. In this case, our sodium-potassium exchange pump is going to pump 3 sodium out of the cell and 2 potassium into the cell. So it's exchanging ions. And this is a process that is going to require energy because we are going to move against our concentration gradients. Now this is something that we have to memorize. 3 sodium are pumped out. 2 potassium are pumped in. So make sure you know the direction of the ions. Again, that sodium goes out, potassium goes in, and you also have to memorize the number. So you need to know 3 sodium pump out and 2 potassium pump in. You will have exam question on this for both our unit and for our departmental final, and you'll continue with this important concept as you move into your next semester of the course. Now the significance here is that because we are pumping our potassium into the cell and our sodium out of the cell, we set up these new concentration gradients where most of the potassium is going to be inside of the cell and most of the sodium in the bottom is going to be outside of the cell. So this picture here is showing we have pumped all of our sodium out and we have pumped all of our potassium in, and so we see this important relationship here. Because it's an unequal exchange, 3 sodium out for 2 potassium in, we see that there's a high concentration of positive charge on the outside of the cell. And so that means anything that has a negative charge inside of the cell, other proteins and other negatively charged ions, they're going to be hanging out right inside the cell membrane because remember, opposite charges attract. And those negative charged proteins and negative ions are attracted to the positive sodium that's been accumulated in the ECF. And so when we measure across the cell, we'll see at rest, a cell is going to have a slight negative charge. And that's another important concept that we do need to memorize. So the resting cell will have a slight negative charge, and that negative charge is a result of our sodium-potassium exchange pump, which is an important example of active transport. The last of our types of diffusion is actually osmosis. Now, osmosis is a special type of diffusion. It's only describing the movement of water across the cell membrane, and so it has its own video, so stay tuned.